So thank you so much to the organizers for inviting me to speak. I'm really excited to tell you a little bit about what we have going on at the P20 Exploratory Center at Vanderbilt University. I'm a fourth-year graduate student who's co-mentored by Drs. Jonathan Schmitz and Maria Hadji-Frangiscu. And today I'm going to talk to you a little bit about a collaboratively developed pipeline for microbial genomes. So I don't think I need to belabor too much with this group, how much of a burden that urinary tract infections cause. But I think what we sometimes underappreciate is how diverse these bacteria that cause urinary tract infections are. So as we all know, about 50% of women will experience at least one episode of a UTI in their lifetime. And the vast majority of these infections are caused by uropathogenic E. coli, about 80% of these infections. However, just the presence of bacteria in the urine or bacteriuria is not just the only indicator of a urinary tract infection. And it's actually a spectrum of clinical presentations, ranging from completely benign asymptomatic bacteriuria where there's no symptoms in the source patient, all the way up to a very severe bacteremia and pyelonephritis infections. And what I hope you can appreciate from this 2017 isolate graph is that we have a wide variety of isolates. Even more recently, Dr. Schmitz and Hadji-Frangiscu are co-directors for a microbial biobank that we have at Vanderbilt called Microbial, which really is going to underline the basis of this pipeline that I'm going to tell you about today. This biobank operates in conjunction with the Vanderbilt Urinary Tract Isolate Repository. And here we bank isolates from urine and now also respiratory isolates for research purposes. One of the main highlights of this biobank is that it's connected to a de-identified patient data for these bacterial isolates. This allows for connecting bacterial genotypes with patient phenotypes. At this point, we've banked tens of thousands of isolates, so we have a really wide and deep data set to work with. Just as a sampling, just from January to September of this year, we've banked over 12,000 urinary isolates, with the vast majority of them being E. coli isolates, next followed up by Klebsiella isolates. So again, what I really want to emphasize here is we are able to really bank a lot of the wild type diversity that's seen causing urinary tract infections. Additionally, this also picks up lesser known and less studied uropathogens such as Acinetobacter, Serratia isolates. So we're also able to leverage this pipeline to look at these lesser studied uropathogens. So I've been working to create a computational pipeline with input from various expertise in order to make the most of the information afforded by this biobank. So really what I'm trying to do is utilize input from a variety of different expertise, again, to glean the most information that we can from this pipeline. So what I've done is I've taken input from basic science researchers, clinical microbiologists, as well as clinicians and physicians actually patient-facing. And what we want to do is make a pipeline that would be useful for all groups and are accessible for information for all three of these groups. Again, with the basic goal of advancing urology research. So what we want to keep in mind here is maybe researchers might want to know what genes or specific alleles of genes are found in these isolates. What's the prevalence of them? What's the prevalence of different MLSTs, sequence types? For clinical microbiology, just basic genomic surveillance of pathogens. Again, looking at the prevalence, maybe even the antibiotic susceptibility profiles of a variety of organisms. And additionally, for clinicians, additional information that could inform patient care down the line. So just to give you a nice 50,000-foot overview of what this pipeline actually looks like. So ideally, what we're doing is we're getting these isolates from the clinical microbiology labs and from Microview where they're already biobanked. And what we're doing is we're sequencing these isolates. And then we're doing some downstream analysis, including genotyping, getting information about the multi-locus sequence type, phylogroup specific for E. coli, but other forms of genotyping, maybe the type of capsule on the bacteria, et cetera. We can then go ahead and unbiasedly annotate these bacteria, find out what types of genes are in these. And finally, we can do even further downstream analysis where we can do some SNP calling to identify differences in genes of interest. We can do association studies and start to really correlate genotype with patient phenotypes. But some of the key features of this pipeline is really to make a really generalizable pipeline for use of bacterial isolates as a whole and keep it modular so other people can use it and apply it to their own research. Additionally, this allows for really high throughput analysis where we're able to sequence and then analyze hundreds, thousands of isolates. And additionally, it provides a really nice framework for genomic surveillance. So if we want to track changes within different bacteria, different virulence factors, we can do that with this pipeline. So I'd like to show you a little bit of what this looks like with real live data. So just as proof of concept, a lot of my thesis work actually works on looking at uropathogenic E. coli. And one of these reasons is because of just how prevalent this pathogen is. Again, it causes up to 80% of urinary tract infections. So it really provides a nice proof of concept for how this pipeline looks and how the data looks. So what I want you to be able to take away from this figure is that E. coli is a very, very diverse uropathogen. I think we always think of it as being in the phylogroups B2 and D, but actually what we're able to see when we start to sample a large quantity of these is we're actually able to pull out E. coli from every single phylogroup. group. So here's a phylogenetic tree that's rooted on the classic strain UTI 89. And what I hope you can appreciate by this inner ring here is that we see colors from all the different phylogroups being represented. Additionally, what I'd like you to pull out from this is disease severity is also not predicted by the phylogroup, and we're able to see diversity in the presence of symptoms or no symptoms in the source patient all around the phylogenetic tree, represented by black and white squares on that outer ring. So taking a deeper look, once we have a snapshot of this diversity, is once we start looking at the virulence factors, it's known that there's no single virulence factor or no single genomic feature that differentiates symptomatic from asymptomatic bacteria. But what I hope you can appreciate from this is it's really truly a checkerboard, and there's a wide variety of carriage of virulence factors within uropathogenic E. coli. So here we have a variety of virulence factors on the y-axis associated with iron uptake, toxins, adhesins, motility, and some others. And what we see is there's really no correlation with the severity of the disease as shown by the bottom x-axis and the presence or absence of these virulence factors. So just to give you a better idea of now what we can do once we actually know what sorts of genes are within these isolates, I'm really excited to show you some super preliminary data that we have where we're now applying genotypes to predicting patient phenotypes. So very recently, we've been able to apply machine learning models to try to predict the presence of symptoms in our source patients. So here is just a very small snapshot of what we're looking at. So what I used was Cover, which is a machine learning algorithm that is publicly available, in order to minimally identify specific k-mers that are associated with your given phenotype. So here I use the presence of symptoms in the source patient as our phenotype. And what we're actually able to pull out are consistent hits associated with two loci. So what I'm showing you here is a loci associated with UTI 89, where there's a pathogenicity island integration site. Additionally, in CFT 073, we find another pathogenicity island integration site where we are getting multiple hits within these loci here. So we're able to start looking and finding genotypes that might inform the patient phenotype, which will impact how we might be able to diagnose in the future the pathogenic potential of a given isolate.
You've got about 1 minute left.
Great, thank you. So finally, I'd like to leave you with one other newer application that we're applying this pipeline to, and that's Pseudomonas aeruginosa. A lot of this work has been done in conjunction with an MSTP student in Dr. Haji Frangiscu's lab, Seth Reisner, where we're isolating Pseudomonas isolates from the urine as well. And just briefly, what I want to show you is we are able to pull out additionally very diverse data very diverse organisms from the urine. So this is just some of the preliminary work where a lot of these are being isolated from catheter-associated UTIs. A large majority of them are hospitalized patients, but we are seeing isolates from both asymptomatic and very severe patients. So we are seeing a nice wide variety of diversity. I've highlighted this red circle to show the more diverse group that's further extended out from the main group. But this work will be more to come in the future. Hopefully Seth will present next year. So just looking into the future very briefly, we can use this computational pipeline to leverage massive amounts of genomic information that's available, not only from internal sources, but also publicly available data sets like NCBI and Patrick. We can utilize clinical microbiology resources to connect bacterial genotypes and patient phenotypes. And finally, this pipeline can truly leverage translational discovery. There's a lot of opportunities to connect researchers with clinicians and vice versa. And it provides a really nice high throughput research pipeline for uropathogens. And finally, I'd just like to acknowledge my labs, my funding, and the variety of institutes at Vanderbilt University that are making this work possible. Thank you.